It's a tapestry woven with shared memories, cherished friendships, and paths that once converged under the same roof. Today, we gather not merely to reconnect, but to celebrate the echoes of laughter, late night conversations, dreams nurtured, and journeys embarked upon. For every corridor once walked, every lesson once learned, and every bond once formed, this moment belongs to you. Good afternoon, everyone. A very warm welcome to each and every one of you, our distinguished faculty, dedicated staff, our incredible students, and most importantly, our beloved alumni. It is a privilege to have you all here today. I am Avantika Harbola, a first year master student in the MIRM family, and it brings me immense joy to welcome you all to this moment where the past and present blend so beautifully. Today is not just an event on the calendar, it is a celebration of stories. Stories of growth, resilience, aspiration, and success that begin within these walls. We gather today for a celebration that is close to all our hearts. Sangam, the alumni meet of the Manipal Institute of Regenerative Medicine. The word Sangam means a confluence, a meeting of rivers, and that is precisely what today is, a meeting of journeys, of memories of the past and the present, all flowing together in this one beautiful moment. Sangam is not just an event, it is the soul of our alumni committee. It is a platform where bonds are renewed, stories are shared, and legacies are celebrated. We are delighted to formally recognize and launch the Sangam Alumni Association Committee, which will serve as the bridge between our institution and its growing family of alumni across the world. As we know, light has always symbolized knowledge and hope. And now, as in tradition, we begin by invoking light, because every great beginning deserves to be lit with purpose and warmth. May I kindly invite our respected Dean, Dr. Jyoti Prasanna, to grace the stage for the lighting of the ceremonial lamp. I would also like to request Dr. Meenal to please join us on the stage. Along with them, I warmly invite Ms. Neha Ji, the President of the Sangam Alumni Association, and Ms. Anuradha, the Vice President of Sangam, to kindly come forward and join us for the ceremonial lighting. That truly sets the tone for everything that follows today. And to carry forward this spirit of celebration, we have some wonderful talent from our own MIRM family. What better way to set the spirit of this celebration than with a little culture and art? First, we have a melodious performance lined up for us by Evelyn Soni. She's currently pursuing a master's here at MIRM.
Hello. सान्द्रानन्दावबोधात्मकमनुपमितम् कालदेशावधिभ्यां निर्मुक्तं नित्यमुक्तं निगमशतसहस्रेण निर्भास्यमानम् अस्पष्टं दृष्टमात्रे पुनरुपुरुषार्थात्मकं ब्रह्मतत्त्वं तत्तावत् भाति साक्षात् गुरुपवनपुरी हन्तभाग्यं जनानाम् ॐ विश्वं सृष्टिच्च विश्वेश्वरा प्रभु आ आ विश्वं सृष्टिच्च विश्वेश्वरा प्रभु विघ्नं विना अरुळीणमे करुणामया निन् कारुण्यदीपकं करुणोदयं पोल विळങ്ങിയിടണേ विघ्नं सृष्टिच्च विश्वेश्वरा प्रभु विघ्नं विना अरुळीणमे भासुरमानसम् ईश्वरक्षेत्रं करुणोदयं पोल विळങ്ങിയിടണേ भासुरमानसम् ईश्वरक्षेत्रं कनिवेरं निन् कृपदया पूजितं विज्ञानामृतं वात्सल्यमायि तन्न विश्वप्रभावति कैतुळुन्नी विज्ञानामृतं वात्सल्यमायि तन्न विश्वप्रभावति कैतुळुन्नी.
Wasn't that beautiful? Thank you so much, Evelyn. What a wonderful voice, and what a perfect way to begin this celebration. And the energy is only going up from here, because next we have a dance performance by Purvi Shivkumar, a PhD scholar at our institute. Purvi, please come on up. Incredible. A big, big round of applause for Purvi, everyone. What grace, what energy. Absolutely stunning. Thank you, Purvi, for that memorable performance. It is a reminder that our institute nurtures not just scientists and researchers, but artists and performers too. And now, with the hearts full and spirits high, we move forward with our program. I would like to respectfully invite our Dean, Dr. Jyoti Prasanna, to deliver the welcome speech on stage. We are honored to have your presence.
First and foremost, I think I should thank our alumni for taking time to come on a Saturday afternoon. That too, when it's so hot. Bangalore is not the same. I think most of you would agree that it's almost like Delhi or, it doesn't feel like Bangalore anymore. So thanks for being there. Thanks, Priyasha. Thanks, Achut, and also Sarvani, who is online with us. And all our young students who are presently here and have organized the event, and some of them who are nearby in NCBS, in STEM, in and around Bangalore, who have made all efforts to come here. And I would not call them as alums because they keep visiting us, be it Ahana, be it Achut, be it Priyasha, or forgive me if I miss some names, but they do, whenever they are in Bangalore, they make sure to come and then tell about themselves and then reminisces whatever fond memories they had from MIRM. So I think it feels nice because as teachers, I think teaching and research is the only profession where you never feel that you're old enough, because with every batch you have a youngster and you feel that you have a baby to nurture, right? So there'll be new babies coming every time with new personalities, with new antics, with new confusion, and of course, their own creativities. So it also helps us evolve with each batch. And we just don't age, but we also reverse age. So that's why you can see some of us, I can say, still young. Uh, so we continue to be young because of you guys. Uh, it's really heartening to see all of you, because especially for me, who has been there from the inception of this institute in 2007, when we started in Manipal Hospital. We were completely unstructured. We just wanted to start a course because there was no manpower available in stem cells. So that was the main motive. It was not to start a big institute or an academic program. And we started with a batch of 30. Meenal and Shagufta. I don't know whether she'll be joining. We are some of the few people who were there from the beginning, where we just had, empty rooms and we had to start everything from scratch with a batch of 30. And I'm very, very happy that some of those 30 people are in fact leading groups, and they're group leaders in it. India and abroad, and they still are in touch with us. Some of them, like Joey, Shatrunjay, Kirti, they keep connecting to us, and you feel very nice when they come back and still remember your classes or whatever they had the feelings for us. So we started with stem cell biology, and then we had seen 3 shifts of institutes, right? From Domlore, then we were asked to pack our bags and empty the building and start everything afresh at Alal Sandra, where we didn't know, there are no eating out places. There was one small shop, right? Anna shop, which was there, and we all used to go there and have. Of course, you should not talk about the hygiene part of it, but all of us used to enjoy that cups of coffee, all of them mixed together, the faculty and the students having it together, right? And then we had the third. So the beautiful campus which you are seeing now was not there before, but then I still cherish the memories. I mean, it was as lively, even if it was in Domlore, and it was as lively even in Alal Sandra, and the group is just expanding. Right, we started with 1 program, and now we have 3 programs, right? 4 programs. The advanced PG diploma was there from beginning, right? Advanced PG diploma started in 2011, but now from 2023-24 onwards, we have masters by research programs in immunology as well as neuroscience. So all these concepts actually somewhere originated from our students. So we feed on our students. I always feel that we feed on our students, and a teacher is as exciting as the students are. So you're the one who make us alive, and I'm not saying it for just this platform. And it's very nice to see some of these people who are up there, doing so well, Achyut, Priyasha, Sarvani, because I can still remember when they started in their classes, the kind of questions they asked, and as confused individuals, because I remember Achyut when he presented a paper, if I'm not wrong, on tendinopathies, and he was talking about tendon regeneration. I was part of it, and we had a discussion as to why tendons can't regenerate as much as other organs, and what could be done. And then, like, he was talking about whether an inflammatory component is involved or not involved, because tendons don't have vasculature. And I still remember those questions. And after the class, because I do take connective tissue classes, and he had come to my cabin, and we had a discussion for whole 30 minutes. Maybe Achyut, you might not remember, but I do remember. And I was like, I want to work on this, ma'am. Like, we should look at mechanical stresses and understand how tendons, it's not just only about vascular and the micronaceous components, but you also have systemic cues which might impact tendon regeneration, and we should look at it. And I'm very interested in it. And I want to work in people who work in biophysics and understand that. And he said that probably I would go to NCBS and look at people who will be doing it. And that's where his journey started in C Camp. And the other reason why I interacted with him more closely was because he was the first person in the institute actually to get a CSIR ranking of 63. And he had given the exam one semester before the actual degree will end. And poor chap, like, he went to NCBS and he wanted to utilize that, but it could not be utilized because 6 months, the I mean, he didn't yet finish his MSc, and CSR was not allowing, and all those things. So there are many things and many stories like that. And then he joined C Camp, then Colin's lab, and then still, what is heartening for me is this: despite all these years and despite so many changes, you can see this, our students finding their way. And 2014, 15, we had this discussion, and in 2026, I see the title of his talk as Muscle Regeneration in Biophysical Cues in Mechanical Stresses. And that is what, sort of heartens me, that somebody has sort of pursued their interests from MSCs with those basic questions and is now a postdoctoral researcher. I think that is very heartening for me. And then coming to the next speaker, Priyasha, she also had been closely, I mean, all of us are in WhatsApp group, and it's very nice that she keeps on sending, she's one person who always keeps sending birthday wishes, and after all these years. And it really means a lot. I remember you had done your, I think, internship in Arjun Guha's lab, right? See, I remember, on lungs. And then after that, you went to ISC in Sachin's lab. And then you were very confused at that time as to whether you would want an academic career or you want to have an industrial career, and how you would do. So I have closely followed your journey. Like, you went into business and then business to academia, and now you're in Mahindra University as a project research scientist. So I think the research bug has not left you, and you've finally found your way. And that is something which is very nice, because Priyasha, I particularly remember we went to BioNeeds. You came with us because in those days, and as of now, we still don't have an animal house. And we were at Alal Chandra campus, and one of my PhD student had to do a orectomy experiment. So that means you have to autoclave everything, go to BioNeeds, which was almost 2 to 3 hours from Domlur, and we all went in Vaishali's car, right? And she volunteered that she will help us. and she wants to see through the whole process. So we had I mean, it was a 3 hours journey, and then there was a traffic jam, there were trucks, and we almost took 6 hours to reach BioNeeds, and finally we learned ovrectomy and did all these. These are the kind of remembrances which, and it was very nice of her, like, to voluntary come help the PhD students, organize stuff out of her own interest. So although she would say that I'm a business person, when she said, I knew she would not sustain that and she'll come back to research, and I can see her as a project research scientist at Mahindra University. So these are the things. And Sarvani, again, we had interacted. She's Meenal student, in fact. Like, Meenal and Sarvani were doing a project together, and they were working on beta islets, right? In Professor Anijat Kumar's lab, and you were the one who were sub-mentoring. And she had discussed that she would want to be in research career. So we have three people, one driven by interest, one who was confused but found her way, and one who was very clear from the very beginning. But out of the three alumni whom you see here, it is very heartening to see that finally they're doing what they like. And I would like to take some undue credit for, not due credit, but maybe undue credit, that somewhere we did maybe something right, that our students have the resilience and the guts to stay there. And of course, in an era of agentic AI, and where all information is available, and you can learn anything. But I think what we try to train in MIRM, you can correct me, and we are always, always open to correcting ourselves. And that's where we are correcting ourselves, and we are reinventing ourselves with each batch. And each batch is very, very different. Very, very different. They have their own problems. They have their own creativities. But what is most enjoyable is how we engage with them. And it's not just that they learn from us, but I feel that we learn from them. Because when Achyut asks a question that is inflammation important, it is a problem statement for me, because a raw question asked in a class comes with a lot of unconditioning, which comes from reading too much of literature. So those questions are like really, really valuable, and it keeps us thinking beyond the classes. And the interactions with students is something which we cherish, we remember. And the reason why we wanted this is, I think I would give full, I don't want to take any credit for this alumni association. It is completely Dr. Meenal's initiative. And she was like, I'm anyway making an Excel sheet of who are all there, where are all there, and they keep coming. So let us have an organized group where, we can interact with each other, where the senior batches can probably share their experiences, because some of you who are there in your final years would be as confused like how they started. And then probably they have found their way. They are maybe less confused, but you are confused in an organized way, maybe about muscle differentiation or something like that, right? So ultimately, it's better you talk to them, because they are your peers. They will tell you what are the journeys which they have traversed and how you can learn from them, and they're there because of that. And when they joined, like, how confused were they? And when we actually started, they were very small batches. Like, there was a batch which was just 7 or 10 students. I don't know how many members, your batch. So I remember all the 7, all the 7 or 8 people in the batch, right? So we never had a batch which was about 30. Sometimes it was 7, it was 14. So we remember all these people by their names. And at that time, I was more involved into teaching than now, where I hardly come, and I used to take stretch of classes. So they had to sort of bear with me, because, I would come late and I'll take for a longer time, and mostly it would be before the lunch hour. And some of them who are interested would come back. In fact, I got a card saying that I'm the most animated teacher, and I still have kept it. So, so each batch has a view about you. It is really nice to see youngsters, making us feel alive. because otherwise teaching seems to be a very, very boring job. And which each batch we have learned. We have learned from our alums, and we continue to learn from them, learn from their journeys as to how they resiliently stood there and did whatever good. And not just praise them, we want to say to all our alums as well as the current batches that we are there for you. We are there for you, even if you have left, and anytime you want any suggestions, any help. I mean, of course, I won't say help, you can help yourself. That is what. But anywhere you want to have a chat with us, you want to talk about your uncertainties, you want to talk about what do you want to do further, we are always, always available. And we want to do it in a very structured way. Thanks to Meenal for that, that we are creating organized events, organized groups, so that, we talk about these things, maybe in an informal setting, get together and help each other. Yeah. And we don't need marketing. In fact, MIRM doesn't have a marketing portfolio which will sort of like take care of our admissions and all. Our admissions have always been taken care of by our own batch of students. And you see the same students coming from the same institutes, because the seniors keep telling. So the legacy sort of continues, and each student is a story, and each student is a story to cherish for at least the faculty. And I can go on forever, but I would like to thank Meenal for that, and we should have more get-togethers like that. I'm open to any kind of ideas and support systems which we can give, so that we can have gatherings like that. Maybe less formal, we can have some sports events where the alums and our present batches can participate. So you can actually look at the calendar when these people come. Some of them, for their abroad, they generally come over December or the Christmas time. So we can sort of shuttle, or we can make our calendar in such a way that they meet. We would like to host some of our alums. They come and talk about their research, talk about their experiences, and we are ready to host them. And maybe one or two people, we can take care of their travel and their coming here too, right? So that the group expands. And this is the first I would like to congratulate Dr. Meenal for putting all this, and the coordination team for putting all this together in a very short notice, and for forcing me to have a budget for that. Yeah, I know I'm there for that, but ultimately it's their blood and sweat. Okay. With this, I'd like to welcome all of you, and anybody who wants to take this podium. I want it to be as informal as possible, where you can talk about what do you think, I mean, how we should better this association. And I won't use the word collaboration because it looks very formal. I would say, how do we strengthen this bond, right? How do we connect the present to the past and the future, right? Yeah. Okay, thank you.
Good afternoon, everyone. My name is Shrida Srivastav. I'm a first year master's student. Now we come to a very momentous moment, one that many of us have been looking forward to. It gives me immense pleasure to announce the formal inauguration of the Sangam Alumni Association Committee, the official body that will represent, connect, and strengthen our alumni network going forward. This committee represents the spirit of giving back, of our alumni choosing to stay connected to this institution and to the generations that follow them. Each member brings with them a very unique story, a unique set of experiences, and an immense amount of passion for this community. I request our Dean, Dr. Jyoti Prasanna, to please honor the committee members with their badges, formally inducting them into their roles. I would now like to invite the following committee members to the stage to receive their badges. So firstly, we have our president, Sneha Ji. Our Vice President, Anuradha Krishnan. Our Treasurer, Pranav P. Nair. Core Committee members, Avantika Harbola, Shravani Zagre, and Pragya. She's out of station, so she couldn't be here. I would also like to call upon Dr. Meenal for a group photo on stage. Thank you. Please join me in congratulating our newly inducted Sangam Alumni Association Committee. We wish them every success and look forward to all that this committee has to achieve. I now request everyone to take their seats. We'll start with the program.
And now we move on to one of the highlights of today's event, our alumni talks. We have the privilege of hearing from three remarkable individuals today, alumni of this very institution, who have gone on to crave extraordinary paths for themselves. To introduce our first speaker, I request Dr. Jyoti Prasanna to please come up on the stage.
I think I had finished his introduction partly during my introduction talk itself, but since it's also a semi-formal event, so I have to do the honors. So Achyut is from our 2014 batch, and he was actually a late entry to our program because he did a BA in biochemistry from Boston University, and he had a 3 to 4 months gap, and that's where he had come as a very confused person that could I actually cope up with the course, and like would I be able to take it up, because the degree was not yet awarded at Boston, right? And it was a last moment induction in our program. So he started his journey as a BA in biochemistry from Boston University. Following that, he joined our institute in 2014 as a master student in the stem cell and regenerative biology program, or flagship program. Actually, we are the first in Asia to start a master's program in stem cell and regenerative medicine, right? So the entire course structure was sort of designed for the need of the industry and the healthcare. And that's where you can see the syllabus to be very atypical. It's not, it doesn't sound like biotechnology, it doesn't sound like genome engineering. You have everything of everywhere, you know, with some amount of developmental biology as well. And so he came as a very confused individual that a first semester is always very, very bad for stem cell biology people, because you have biomolecules. Most of you find it very hard to sort of get through biomolecules. And then he was like, you know, how can I tackle with the course, because he was 3 to 4 months late. But despite that, he picked up very fast, and is in fact the topper of that batch, if I can say so. And I thoroughly enjoyed, especially it was a very, very small batch, but the most interactive batch, one of the most interactive batch, Priya Shah, Achyut, all of you, right? Then he went to C Camp, where he worked with Dr. Tasleem and then with Dr. Colin as a project assistant, and that is where the CSIR fiasco happened. But then he navigated his way through. He applied for PhD positions abroad, and then he came one day and he told that finally I got a PhD position in France in a developmental biology institute, where he has consistently worked on how myogenic differentiation happens and how mechanical stresses and cues impact the differentiation. I think think he's going to show some beautiful data there. And Achit, I want to say that we all suffered for the imaging, but now in the new campus, we have a good confocal imaging. So probably, we used to go to NCBS and CCAM to get even the basic pictures, right? Basic confocal pictures. But now the institute has evolved. So it's very nice that the students have done so well for themselves, even when the facilities were not so much. And it is on us, all our faculty, to give them the best and the bestest to our alumni and all our students. So you can see how he has resiliently, consistently pursued his passion for muscle research, looking at how, because for tendinopathies, like he used to discuss a lot about it during that time, the isometric training and all, the only ways help can be provided. It sometimes takes weeks to months to heal a tendon because it doesn't have vasculature. So whether inducing inflammation is beneficial, not beneficial, and maybe that's the reason he took up a position in Dr. Colin's lab, because Colin works on inflammation and stem cell fate choices. So you can see that his journey has been a very calculated choice. It has not been a choice. I'm not saying he has arrived or he has not arrived, but I'm talking about how he pursued his passion and how his choices were dependent upon the initial questions he wanted to ask. And he has consistently pursued this interest from past 12 years. Right, and he has published really well. He has a good paper in eLife. I have not gone through it, but it is on iPSC-derived muscle fibers and how tension-driven multiscale organization affects myogenic differentiation. It's very complicated, but I think it means how your spatio-physical cues could impact differentiation choices. So he has published well, and we are here to listen to him, listen to his journey and his play with muscles, right? And he's going to show a lot of videos and color. I can see that. Yeah, welcome, Achit. Yeah.
Hello, everyone. Firstly, thank you, Jyoti ma'am, for that wonderful introduction. Really. I know this is supposed to be a formal event, but anyone who knows me knows I don't do formal very well, as evidenced. So, keeping in mind, please feel free to jump in, interrupt me, ask me a question at any point in time. I would much rather make it an active discussion than a boring presentation. And if anyone falls asleep, just don't snore. Apart from that, go for it. So, today I want to talk about some of the work I have been doing in France for the past 6 or so years. I've changed the title a little bit. Maybe it's less interesting for some, maybe it's more, but I'll try to keep it interesting. So I've been working on muscle development, and one of the key focuses that we've had is to try to understand what is the interaction between biology and physics in this muscle development. And this is a little complicated maybe, but the work is being done at IBDM, the Institute for Developmental Biology in Marseille, and also with the Laboratory for Adhesion and Inflammation. So, yeah, with Aix-Marseille University. So without further ado, let's start. Why do we care about muscles? It allows people to do stuff like this, and this, and this. And despite how drastically different all three of these movements seem to be, they're powered by a relatively simple conserved mechanism. In mammals, it looks something like this. So you have the muscle itself, which is connected to two skeletal elements via tendons. In this case, it's the bone. And the contraction of the muscle moves the bones in relation to each other, which is what we perceive as movement. This is quite complicated. Let's make it a little bit more complicated. A skeletal muscle is a hierarchical tissue which is a bundle of bundles. What do I mean by that? You have fascia, which are all rolled into one muscle bundle. And within each muscle fiber bundle, you have multiple myofibers. Every muscle cell or myofiber is composed of these elastic strings, which are called myofibrils. And the composition of the myofibril is made up of these sarcomeres in series. And the sarcomere is the smallest contractile unit of a muscle. To take a slightly deeper look at the sarcomere, this is the sarcomere in some of its glory. The most important proteins are what I have marked out. So what I've highlighted here in green is the Z disc, which anchors the edges of the sarcomere. You have the actin filaments that run in towards the center. You have the myosin filaments that start from the center, moving outwards. And this stringy, colorful protein is what we call titin, which basically anchors the sarcomere on the Z disc at the C terminus and the N terminus and the center of the sarcomere at its C terminus. How does such a complex tissue, such a complex mechanism arise in vivo? This is a simplified view of myogenesis. You start out with mesodermal progenitors, which give rise to myogenic precursors, as well as precursors for cartilage and tendon cells. And this complex 3D milieu is what tells these cells how they should elongate and grow. And eventually, they interact with tendon cells, with the motor axon, with bones and cartilage to produce what is the final muscle. Given how complex this looks, how can we simplify it to study it in a lab? What we use is an in vitro 2D culture system with no external biological cues. We use the same biological developmental pattern. We take iPSCs, differentiate them into mesodermal progenitors, make them into myogenic precursors. We then isolate these satellite-like cells and then grow them on dishes to form myofibers. So how does this actually look like in reality? Also, what are the questions that we want to ask? What are the inherent properties of these myogenic cells? If we remove the 3D environment, the cues provided by the biology, what do these cells want to do just by themselves? How do they differentiate in the absence of biochemical cues? And what role does tension and other mechanical cues play in this differentiation pathway? So, when we differentiated these satellite-like cells, we see a pretty nice developmental pattern over the course of 2 weeks. They start out as small, flat, fibroblast-like cells. And then over the course of the first week, they elongate. And then over the course of the second week, they start to thicken and start to show nice sarcomeric patterns, which I will show you later. So when we quantified this data, we see a very curious behavior. So there is a very dramatic increase in the length of the cells for the first 7 days, a dramatic decrease in the width of the cells for the first 7 days, and a pretty rapid increase in the nuclear number for the first 7 days, which then continues to grow, continue to grow, but not at the same rate. So there's some sort of a biphasic behavior that we are observing. To look at it from a different context, by zooming out and looking at it from the tissue scale, what we see here is the entire culture system that we have in this pizza-shaped insert. And you can see a multitude of myofibers, very small, very tiny, and there doesn't seem to be much of a pattern. So this is a pseudo-color representation of the orientation. So the color of the cells is based on what orientation, what angle they're in. And as you can appreciate, over the course of differentiation, these bundles seem to grow larger and they seem to be better organized. We found this quite interesting. Somehow, they're self-organizing without biochemical cues being added into the system. So what we did from these simple confocal images was we tried to trace different myofiber bundles over the developmental time course. And the red dots that you can see are what we identified as foci, or regions where multiple myofiber bundles end. And what is quite evident is the bundle numbers are lower. As you age, the bundle sizes grow, and the number of foci reduces. This is something that we ended up quantifying. And again, similar to on the cellular scale, we see a similar biphasic phenomena where the bundle number decreases dramatically for the first 7 days and then seems to stabilize. The bundle area increases dramatically over the first 7 days and then seems to stabilize. And the foci number follows a similar pattern as the bundle number. So this is all well and good, when you're seeing it as time frames in different samples. Is it something that is also appreciable across a real live culture? So for that, we created this little movie where you can see that there are multiple bundles that are actively migrating, elongating, and merging together. So just to reiterate the point, if we focus at the bottom left, you start out with 4 smaller bundles with about 7 foci between them. And as you progress from day 3 to day 4 and then day 5, these bundles and cells interact by meeting each other, and as they meet their neighbors, they want to align with them. They begin to slide along each other, and that's how these bundles seem to be merging. So we've seen an interesting self-organization behavior on the cellular level and on the tissue scale. What's happening on the molecular scale? Because a muscle isn't a muscle if it doesn't have good sarcomeres. So this again is our developmental time frame. What we're looking at is the N-terminus of titin here. And we have categorized these cells into 4 classes. We have the myotubes, the immature, transitional, and mature myofibers. So this classification was done on the basis of the expression levels and localizations for titin and myosin. What do I mean by that? So you can see that in the myotube stage, you have pretty bad expression levels for titin. That seems to improve when you get to the immature myofiber stage. And then at the transitional and mature stage is where you start to see a very nice periodic pattern emerging in this expression of titin. This indicates to us that what we are seeing at the bottom is a real sarcomere and not just some random aggregation of proteins. But we are humans, we have our inherent biases. So what we wanted to do for the paper was see if we can computationally find this periodicity and remove any human bias involved. Is there an actual difference on a computational level between what we see at the immature myofiber stage versus the mature myofiber? So to do this, we employed something called an autocorrelation function. I will not bore you with equations and mathematics. I'm just going to try to explain it as simply as I can. We take a small bit of the existing pattern and compare it against itself. So for example, from the pattern that we have on the left, I've taken a small bit of it that I've highlighted in yellow and then compared it to itself. So when you compare a pattern against itself, you have a perfect correlation, which means you start with a correlation coefficient of 1. Then you move that pattern slightly. Now the correlation reduces. So you now have a low correlation, which means the coefficient has gone down to about 2. move that pattern further. Now you have anti-correlation, which means your correlation coefficient is in the negatives. Slide that pattern further, and you start to see an increase in the correlation. So this is a very simplified understanding of autocorrelation. And what this would allow us to do is basically measure when and how we have these patterns emerging in our cell cultures. So when we took individual myofibers across all of these days, we see that day 1, 2, 3, there isn't really any pattern. And then slowly around day 4 and 5 is where pattern starts to emerge. And then it gets really strong at the later time points at the end of the second week. And when we plot individual, the patterns from individual cells, we see that at day 2, 3, there's not really any pattern there. And then there's a stronger pattern at day 15, day 12, day 9. But in that middle portion, there is a lot of heterogeneity. So we went back to our 4 categories, irrespective of which time point they were selected from. And then we plotted the autocorrelation. And this one gives us a much stronger result, where you have absolutely no periodicity at the myotube stage. You start to see periodic patterns emerging at the transitional myofiber level. And then it obviously becomes more mature with the mature cells. This is just to prove that I'm not bullshitting you. So what this analysis shows us is that the manual myofiber classification is supported by an unbiased computational test. So this is just to say that our eyes aren't deceiving us. And what we see here is a real pattern which is correlated by a machine. So now we come to the biophysics portion. Is there any tension in the muscles? To test this, we cut individual cells with a laser. Perhaps one of the coolest experiments I had the pleasure of doing. It was almost like Star Wars. Pew. So we cut these cells, and what we're looking at is the recoil. If cells are under tension, anything under tension, if you cut it in the middle, it'll snap. And we want to measure that snap and see if it can tell us something about how much tension these cells exist under. So what you can see here is the red line is where we cut the cells. The green line is the the blue line is the first second after the cut. And the maroon line at the bottom is 5 seconds after the cut. And what we want to do is plot how much retraction happens in that first second, because we want to remove the effect of secondary movements because of myosin or calcium fluxes. So we don't really consider what's happening after the first second of the cut. And clearly what is visible is that the slope of the retraction curves increases up till you reach the transitional level, which means that tension, number 1, is present in the cells even before you have sarcomeric patterns, which indicates to us that as soon as a cell is elongated, there is already tension in the system. When you start adding the contractile machinery, and as that machinery becomes more mature, you start to have an increase in the amount of tension. But is tension actually critical for the maintenance of the sarcomeric pattern, or is it just a random byproduct? To look at that, we have some live imaging data. And we were lucky enough to basically capture a cell that lost its adhesion at the end. And when it loses its grip, it again retracts. And what you can see is the retraction is followed by a loss of the sarcomeric pattern. So tension increases as you have increasing maturity of the sarcomeric pattern, but also without the tension, that pattern disintegrates. So these two things have an interplay, a feedback loop between each other. Another thing, when you're running experiments for 2 to 3 weeks and you have cells detaching after about 15 days, it's like your baby dies. So we wanted to figure out, is there some way that we can improve the, the, the attachment of these cells to our culture system? So to do that, we stuck with the standard culture format for the first 7 days, and then we added a second layer of matrigel on top of them, so that these cells are now surrounded in 3 dimensions by an ECM layer. Hopefully, this should give them enough ECM and adhesion signals to not just snap off whenever they want to. Did we succeed? Kind of. So at the top, you can see the time frames in pure 2D, and at the bottom, it's the same time frames, but in the 3D or the pseudo 3D cultures. And what I've highlighted in the white arrows are empty spots where the cells have retracted and there's nothing there. And then in the darker pink arrow, you have dark regions where the cells have retracted to, and basically you have a clump of cells. These clumps become larger at day 21, and these empty spaces become larger as well. And day 40, this is something I made just to show you guys for this presentation, because usually at day 40 we have nothing. We have an empty dish. So this is kind of what it looks like. Very large empty gaps with clusters of cells just hanging out, holding on for dear life. Compared that to the sandwich or the pseudo 3D cells, they seem to be happy up to day 40, and I have cultured them even up to day 60 for 2 months, and they were fine. They were in fact better than fine, because this is something that we started observing. Hello. There we go. So we actually started to see twitches in our culture system. There we go. So my boss kept asking me for 4 years of my PhD, are these cells contractile? Do they move? Do they contract? Do they twitch? And for 4 years, I kept telling him, they do, but I can't show you, because by the time I turn on the microscope and capture an image, they're done twitching. And with these pseudo 3D cultures, this was the first time we had sustained twitching, which allowed me to finally prove to my boss that I wasn't just talking nonsense. So what this shows us is that the 3D anchoring supports spontaneous twitching, which hints at a higher level of maturity. So now the question that we need to ask is, how does modulating internal tension impact muscle differentiation? Because we know that the two things are linked. The simplest experiment that we could think about doing was simply altering the seeding density. If our hypothesis was correct, then as the cells bundle, that bundling allows them to feel a global tension around them rather than the tension just felt by a single cell. And this global tension can then add to the impetus to differentiate. So if we remove that impetus by removing the seeding density or reducing the seeding density, it should impact the cell differentiation. And similarly, by increasing the seeding density, we should push these cells faster. And that is what we saw. This is images from day 3 of differentiation, where we seeded 2,500 cells per centimeter square, 5,000 cells per centimeter square, 10 and 20,000 cells per centimeter square. The 10,000 is our standard culture seeding density. And what is very visible is that this looks like a time course just by itself, where even though they're all from the same age, they display dramatically different phenotypes and morphologies. So at the 20K level, they've formed very mature bundles. They're all, nicely organized. And what was even more surprising to us was, even on the molecular scale, they actually started forming sarcomeric patterns at day 3, which was, for me, unheard of. So the bundling accelerates myofiber growth and sarcomere organization, and tension has something to do with this. So this brings me to the model that we propose in the paper, which is, you start out with small flat cells, they meet each other, they greet their neighbors, and then if their neighbors are aligned with them, they want to grow together. As they grow together, the amount of tension increases, which then activates the cellular machinery that builds these sarcomeric proteins. At about day 7, they reach some sort of a critical tension level, which then makes them stabilize and also start building mature sarcomeres. And at the end of 2 weeks, they formed pretty stable bundles that don't really move too much. They're not very dynamic at this stage. And we also see very nice focal adhesion patterns at the ends of these myofibers. So far it's been a lot of biology, not so much of physics. Let me correct that. Now we're entering the part which is a lot more unexplored data. So the first thing that we wanted to test in terms of physical properties that can be manipulated is how does altering the substrate stiffness affect the muscle differentiation? Why do we care about this question? Because normally we grow our cells on a polystyrene petri dish or a plastic or glass petri dish, which has a stiffness that is measured in gigapascal, something that is only similar to bone. In vivo, you have a muscle stiffness range that is somewhere between 10,000 to 100,000 megapascal. Sorry, pascal. So we are many orders of magnitude higher when we grow them on these plastic dishes. How does that impact their behavior? This is what I wanted to look at. So we start out with our standard cover slip bottom dishes. We add PDMS, which is a soft elastomer, which we can cut into different shapes. And then we stick our standard culture well on top. So now our system looks something like this. We then coat the PDMS with polydopamine. PDMS has this nasty property that it's not very biocompatible. It's very hydrophobic. So to counter that, we need polydopamine treatment because that actually is a very nice natural glue. We then coat the cultures with Matrigel and then seed our cells on top. So I did this experiment with many different varieties of PDMS. We can alter the stiffness of the PDMS. So we have the plastic ones at the top left. We went as soft as 40 kilopascal at the bottom right. And we saw a very interesting behavior emerge, where at the 4 megapascal, they seem amazingly well organized, bundles that we have never seen, this large before. And then for some reason, at 40 kilopascal, which is in the in vivo range, they seem to be similar to plastic. So we were really confused about what was happening. I spent 1 year working on this experiment, and the final thing that I can tell you is so far, substrate stiffness might have an impact on the myofiber length and bundle dimensions. But never get excited with your first positive experiment, because after 1 year, we realized that everything that we had observed was artifactual. And so we basically had to start from zero with this experiment. So while I was wasting time for a year, I was also working on a different experiment. Everything that we've seen so far, we have nice myofiber bundles, but they aren't really aligned the way they are in humans. We have very organized muscles. Without that, we would not be moving the way we do. So how can we convert what we have, this random orientation of muscles here, into something that is more similar to what we have in our bodies? Can constraining them within a certain shape affect how they differentiate? So again, to test this, I used PDMS, cut it into different shapes. So we have a rectangle, we have a flattened hexagon, and we have some ellipses. And we had some interesting results. The rectangle doesn't really work so well. The ellipse is interesting, but I don't consider it highly successful. But the flattened hexagon gave us some very nice results. So yes, the cells are mechanosensitive and they can feel what is around them and adapt to what is around them. So you can actually somehow train these muscles to behave in a certain way by how you condition them in their environment. So this is all well and good, but the coup de grace is, can we directly modulate mechanical tension within these myofiber bundles, and how can that affect these cells? So to do that, we designed an in-house cell stretcher. What you can see on the left is a top-down view. So you have the motor here. This dark gray bit is where the cells are cultured, and the light gray here is the chamber, and this light gray is the arm that can translate back and forth while the cells are in culture. And on the right, you have a cross-sectional view. So again, this area is where you would culture the cells, and this is accessible to microscopy, so you can do live imaging, or at least that's the goal. So does it work? Yeah. This is the first prototype that we designed, and as you can see, we can actually program different ways, different modalities of stretching. You can have cyclic stretching, you can have linear stretching, you can have ramp stretching. So not bad, it works so far. How do these cells actually respond to this external stretching? So what we did was we stretched cells from day 3 to day 9 in a cyclic manner for 0.2 hertz, which is one entire cycle in about 5 seconds. And the maximum stretch was 20%, because beyond that it is non-physiological. And we got some very interesting preliminary results. So in the unstretched condition, as you can see, you have myofiber bundles, but they are seemingly arranged quite randomly. Compared to that, when you look at the stretched cells, they all seem to be parallel to the direction of the stretch. So these cells feel the stretch and then reorient themselves even after day 3, when they're still in that dynamic stage. And when we look at them on the molecular level, day 9 in the unstretched, you do have sarcomeres, but they're still in that transitional stage and where they're not super clean. But compared to that, the day 9 stretch samples, you have very nice sarcomeric patterns that we've observed. So this tells us that the external stretch has 2 effects so far. It aligns these cells on a global tissue level, and also on the molecular level, it seems to be accelerating the formation of sarcomeres. So far, what we've done over the last 6 years is we've shown that myofibers have an inherent capacity to elongate to a stable length. Myofibers self-organize into bundles, so they have an inherent capacity for this self-organization without external cues. These sarcomeric patterns emerge around the same time as you have stabilization of the myofiber lengths. So these 2 things are, or these 3 things are all, connected somehow. And potentially that connection is tension, which is acting as a global cue across the cellular, the macro tissue level, as well as the molecular scale. And other mechanical cues such as substrate stiffness and substrate shape all can also impact and educate these myofibers and their differentiation. So with that, I want to thank both my teams. So on the left, you have my team at IBDM, which is led by Frank Schnorrer, who's here. And I've had the pleasure of working with Tian for the entirety of my time here. And we have 2 new PhD students, Leila here and Roma at the back, who are essentially continuing the work that I started with the pseudo 3D and the substrate stiffnesses. And on the right, we have the large team at LAI, and this was my second thesis advisor, Olivier Theodoli. And I cannot forget to thank the engineer that I have worked with for the past 2 years, who's helped me building, designing, and testing the device, Fabrice Desolis. So with that, I thank you for your attention.
Thank you so much, sir. That was a truly insightful talk. And now we open the floor for a question and answer session. This is your chance, students and teachers alike. If you have a question, please raise your hand and we will bring the mic to you.
Okay.
Yeah. Have you ever tried culturing these cells on a fiber, nanofibers, or aligned instead of having a rectangle ones or a square shape, anything which is a fiber like, so that the cells can align themselves in that?
This is something that I have read about many times, but we have not actually performed these experiments ourselves. So the lab in LAI, they have an expertise in bioprinting, so they can actually print ECM in specific patterns. I tested this at some point early in my PhD, and we had a massive contamination issue. So I did not proceed with that experiment any
I see.
further. But yeah, this would be a logical way of doing it. From the results that I have seen in the literature so far, one of the downsides of patterning the substrate itself is that it forces the cells essentially to grow in a parallel manner. So they don't really form foci. You have individual myofibers that align, but they don't form a unit, they don't form a bundle. So I think that is a potential downside, and that is one of the reasons why we haven't pursued it. But
I see.
yeah, it is something that.
But you said when you have your substrates at 40 kilopascals, it behaved similar to tissue culture plastic.
Yes.
So why do you think so?
So as I mentioned, almost everything that I realized after 9 months or a year was that it was artifactual. So the aligning or the alignment that we saw was probably because I was aligning the matrigel that the cells were seated on, and that somehow that topology
Exactly.
cue is what gave the cells that
So,
orientation.
so my question goes back, like the PDMS, you coated it with a few things, right?
Yes.
So probably is it because of that?
Yeah.
So it's you're not able to get the right stiffness. You don't know what's exactly stiffness when you coat the.
So we have
These
an idea of
substrates.
what the stiffness is, but we don't know what the macro topology is. So when we add the matrigel, because of the hydrophobicity of the PDMS, you sometimes have bubbles at the edges where the matrigel does not coat completely. So with those samples, you have to manually go in with a pipette tip and clear out those bubbles. And when you do that, you are kind of scoring the matrigel and creating some artificial pattern that these cells somehow read and recognize and then grow accordingly. So yeah, there is some macro topology happening. We've measured the actual stiffness of the PDMS without any coating, the PDMS with the polydopamine, as well as with the matrigel with AFM. So we know what the stiffnesses are. And the stiffness I mentioned is not of the native PDMS, it's the entire stack.
I see. Okay.
I was curious as to how this ECM is in vivo at the same stage of development,
Right.
and why have you not encompassed that component in your experiments? Because that can be deepithalized in vivo tissue in itself that you can embed your cells in,
True.
right?
So a very fascinating experiment I read about 2 years ago is French people obsessed with wine and grapes. They decellularized grapes and then added muscle cells into the grape and looked at how the cells behaved in this muscle grape jelly thing. And it's fascinating. There is some collagen, some lignin, some ECM proteins that are in the grape, and that informed the cells in their orientation. So you're absolutely right. What would be important is to look at what the ECM is like at those early stages of differentiation. Unfortunately, we're talking about humans, embryonic development. So actually measuring and testing what the ECM is very difficult. There is some RNA sequencing data which shows a very specific pattern of ECM proteins. You have waves of fibronectin and then laminin and then different levels of collagen, 1, 4, 14, 18, so on. So
It's in mouse,
yeah.
I suppose in animals.
Yes. So there is a very specific ECM waves, and these cells are not just static individuals. They actually remodel the ECM as well. So the matrigel that we started out with at day 0 is not the same as what we have at day 15. There is a lot of remodeling happening there, and they're also laying down their own matrix. So one of the reasons why we did not really, go too deep into the ECM components was that we wanted to keep our system as simple as possible. A 2D system where it's easy to visualize would be, the most accessible when you start out. And then once you understand the characteristics, you can then take it to, 3D or pseudo 3D and then make it a little bit more complicated. But the myofibers starting from IPSCs was relatively new. So just for the sake of simplicity and not to have an 8-year PhD, we kind of avoided that.
And have you considered co-culturing with relative mesenchymal cells in the sense of during developmental stage, the cell's mesenchymal or connective tissue is different. The cell is
Absolutely.
different from mature cells.
Yes.
So if you combine the two, what do you think will be the
So one thing I didn't mention is that our cells, our culture system is not a homogeneous muscle culture system. It is heterogeneous because we're starting out with the iPSCs. We actually have a whole bunch of cells, and when we did RNA-seq on this, we did see, mesodermal cells that were non-myogenic. So potentially satellite cells, fibroblasts. We did see some amount of tendon-like proteins, genes being expressed. So it is a heterogeneous culture system, which in a way is good and in a way is bad. We tried to see if the foci that we observed was the result of localizing tendon cells in that region, which then kind of gives it a cue, and that doesn't seem to be the case. So for the most part, it's just muscle cells. And we have considered co-cultures. Co-cultures with motor neurons is something that our collaborators at Harvard have done, and they have produced amazing results. We are interested in doing co-cultures with tendon cells, and I am now kind of building a collaboration with a team in Netherlands who are experts in biomaterials. And this would be potentially something that we explore in the next 6 months or maybe the next PhD student.
It's maybe a question which comes from my naiveness of understanding muscle biology. But do you think the topological cues will affect the decision whether they would want to go to a contractile myogenic stage versus a synthetic? I know that when the ECM changes, especially for smooth muscle cells, you have the change between contractile smooth muscle stage to a synthetic smooth muscle stage. So these are two different phases of myogenic functionality, which is very much dependent upon the ECM cues and the topology. For example, I'm talking from in context of angiogenesis, which we are working on, where the smooth muscles actually cover the blood vessels. Okay, the injury cues actually decide their shifting between the contractile stage to the synthetic stage. So, do you think your topological cues and stresses help in this transition?
So I think the simplest answer is I don't think skeletal muscles behave exactly the same as smooth muscles. And I don't think there is necessarily a synthetic stage as such. They might be slightly less contractile, but I think it's more a functionality of maturity rather than the ECM cues. ECM plays a role in terms of adhesion and patterning. And we have not found any cues as such that say that ECM plays a role in function or contractileness. So again, at the start of my PhD, I did play around with different ECMs because, as you may know, Matrigel is an animal-derived product. It's not, a very consistent thing. With every batch, you have variations in the amounts of certain proteins, the amount of certain growth factors. And trying to make a minimalist system that is reproducible, we wanted to work with something that is more defined. So I tested the myofiber differentiation on pure laminin, pure fibronectin, pure collagen 1, and then different combinations of these three. And the end result that I found is that Matrigel and the components that look the most like matrigel work best. So yeah, at the end of the day, we didn't really see a functional difference in the cells among these different ECMs.
Thank you for those wonderful questions and for such thoughtful answers. It is now my honor to invite our Dean back on stage to present a token of appreciation to Dr. Achyut Acharya in recognition of their time, their wisdom, and their generosity in being here with us today. Thank you, ma'am. Thank you once again, Dr. Achyut. We are so grateful. Moving on, I now invite Dr. Meenal to the stage to introduce our second speaker for the day.
Thank you, Avantika. It's a great pleasure to introduce our second speaker of the day for Sangam Meet 2026. To begin with Achyut and then Priyasha. Great memories of curious Priyasha, I call her that, because she had like questions all the time and never ends. and then she pursued it, like Dr. Jyoti said. I don't want to extend to what Dr. Jyoti has said about Priyasha. She, really lit up the study, how the classes were going on, and some bright light to the class because of her questions. And therefore, like how Dr. Jyoti said, she pursued it. She went after her curiosity and is now in Mahindra University as a research associate, and she pursued her PhD in LV Prasad here in Hyderabad, and her work of interest was in the role of antimicrobial peptides in bacterial keratinitis using combination of molecular biology, cell biology, microbiology, and immunological techniques. And from earlier to that, she was a project fellow, junior research fellow, and to add to it, she is an SRF, a senior research fellow during her PhD, and has multiple publications in the topic she has worked on in her PhD. So I want to, very happy that you came, and want to welcome her to a session of career orientation for our students. Welcome, Priyasha.
Thank you, ma'am, for actually inviting me for this. Hi, everyone. So I actually don't know from where, what to start with. So when Dr. Meenal actually called me probably 3 weeks back, at that point we were still planning, like me and Achyut were planning to come to Bangalore for just a reunion. And then she told me that, we're planning to have this alumni meet, and would it be possible for you to come down? I said, we are planning to. It would be great if it could happen between 11th to 13th of April, because that's when we both would be here. And that's how it started. And since then, so it's like, I'm blank being at this stage again. But first and foremost, I need someone to just plug in this. Okay, so I started, you guys remember the first day of your MSc? You had your entire life sorted. MSc, PhD, postdoc, faculty, research, complete world peace, right? I was kind of same. Like the experiment that Achyut showed, the laser beam cutting through. I had my goals very straight, but they were not aligning with what was actually happening in my life and with how my mindset was going with everything that was around. So I started with my master's over here in regenerative medicine, stem cells. Very fascinating, very interesting. Prior to that, I did my bachelor's in genetics. So I was always, from childhood, I was very curious about this condition called keloids, which was basically over a scarred healing, right? So it has a genetic component to it, which runs in my family. So that was the major reason why I stepped into science, and I wanted to become an MBBS doctor. I think most of us wanted to become MBBS doctor, but then we shifted to, let's do doctorate. DR is still the same. So then I shifted to regenerative medicine after doing genetics. And after that master's stint, I realized that, okay, there's something still missing. I do not know what to do. And that's when the entire confusion and the reality hits that, you know, whatever goals, aspirations, and dreams you have, that always does not align with the real time scenario that you are in after you have graduated from your master's. So I'm going to stand here, not, both Dr. Jyoti and Dr. Meenal told a lot of things about me. They sound very successful, fascinating. Even I'm very intimidated by all the compliments I got today. I feel as if, when you sit there and you see an alumni presenting here, you feel, oh my God, they would have achieved so much. I felt the same hearing all the talks they told about me today. So thanks again for saying such lovely words. I do not have a very scientific presentation today because Achut was in charge of that. But what I'm going to share with you is a very informal chat and a kind of a story. Like as we said in the beginning of this alumni meet, that every student here has a different story to tell. Every path is very different from your batchmate, your senior, your junior, your peers, everyone. So I'm going to talk about my unscripted journey where I was navigating through life, career, and different challenges altogether. This is what my life was after MSc. The day I realized I submitted my master's dissertation thesis, okay, we are postgraduate students, now what? Every one of my batchmate already had a position when we were about to get our degree, except me. Not that I was a dumb student. I had good marks, by the way, really good marks, okay. But I did not have any position in my hand. I was trying, but there were a lot of rejections. First lesson, post-graduation, after PG, after masters, it's not about the success. The entire journey does not teach you about what you're supposed to do. It teaches you what you can do when you have nothing to do, right? So this is where it started beginning. I joined Dr. JP Ma'am's lab as a volunteer. I requested her that, no, I will join as a volunteer in your lab. I'll do whatever it is. I need some experience. I need to work because I can't sit idle in my house, right? I can't keep waiting because all of my other batchmates are working. So then I joined her lab. I learned a lot of surgical, techniques during that point. And then, before that, so this is 2013 when we joined. That was our batch. It's only me and Achut who are here right now. I'm sure a lot of other people also wanted to join, but they could not for certain reasons. 2015, graduated with master's degree in hand from Manipal University. Great. The blank story starts again. The wheel of reinventing yourself and thinking that whatever we were thinking were dreams of a child, and right now it doesn't fit to the professional world at all. Then I joined JP Ma'am's lab. While working there, I kept on, and she always supported me that, Priyasha, if you get somewhere else, if the work is interesting to you, you should go for it. It's okay. You can tell me no. You can stop working in my lab. You can go and pursue your dreams. Because she knew my dreams were all very much chaotic. Like today I wanted to do this, tomorrow I wanted to do that. I don't know, I wanted to do something, but didn't know what would purse. So yeah, I can see a lot of people agreeing that yes. So then this was my lab in InSTEM. So in 2016, I joined InSTEM. I was working under Dr. Koichi Hasegawa, and we were working on induced pluripotent stem cells differentiation. It was a multicentric malaria project where we were trying to establish a diagnostic, a liver stage diagnostic tool to check different malarial drugs. So during this 2-year project, I gained like I was doing microdissections on mosquito larvae. Like we used to take out the salivary glands, take the sporozoites, and then infect the cells. We had malarial patient blood, differentiated, like made iPSCs, differentiated them into hepatocytes, tested these sporozoites. It was amazing. 2 years, I still don't know what I wanted to do after that part. The project got over, the lab dissolved. I was still questioning what am I supposed to do. I remember calling at every stage of this entire slide. I remember calling JP ma'am and messaging her, ma'am, तो ऐसा हुआ है and अब मुझे नहीं पता क्या करना है. Do you have anything? This has been my constant question to JP ma'am. And believe me when I say this, she's always been very supportive and she's like, oh, you can try this, maybe you can do this, maybe you can do this. Then I joined ISC in 2017. This was the lab back there. I was basically recruited there to establish the stem cell facility, which the PhD students would be using to study arrangement of spindle fibers during mitosis. I have no clue what the data was, by the way, because my role started with establishing the stem cell facility and ended within 4 months. I established it, resigned, went underground for 2 months. Called up JP ma'am again. Ma'am, क्या कर सकते हैं अभी? Ma'am, do you have a position? Very sweetly, as usual, for the past 5 years, she told me again, Priyasha, why don't you try at LV Prasad, Hyderabad? I have a person over there, and she's interested in going with iPSCs. एक बार just drop a mail and try. Fine. JP ma'am ने बोला है mail कर देते फिर हो जाएगा सब कुछ. Dropped a mail to Dr. Indumati, who's in LVPI. Right now she shifted to NCBS, by the way. I think she shifted in 2024, 2025, I'm not sure, but yeah. So I wrote to her that I'm interested, and this is my background with stem cells and iPSC differentiations and this and this and this. Very fascinating as it sounded. I had 2 Zoom calls with her. I was still in Bangalore and they were in Hyderabad. 2 Zoom calls over 1 hour each. She asked me about my project that I was working on. I told her, she told me that, okay, we want to do iPSCs from retinoblastoma patients to make a model system and to study the genetic abnormalities between it. I was like, okay, cool, fine. Why don't you come down and just visit the lab once? I was like, okay. Booked my tickets, went down to Hyderabad, saw the lab around, and at the end of the day, she's handing me my joining letter. And I'm still confused. It's not that I told her that, okay, yes, I'm going to take this position. I just look at the joining letter and it's dated 1 week after that particular day of when I was visiting it. I called up my parents. I'm like, Hyderabad में मिल रहा है, PhD position भी है, क्या करना है? Somehow, I came back to Bangalore, packed my bags. Next week, shifted to Hyderabad. I joined LVPI. But things don't go as planned. 2017, I joined LVPI. I left the lab after 6 months. I joined another lab in LVPI in 2018. This lab was in a biochemistry department. I used to hate biochemistry, by the way. Hated biochemistry. I still do not remember the structures of amino acids. Please do not even dare ask me those questions. But fine. The lab and the project had got nothing to do with stem cells. She told me this has got nothing to do with stem cells. It's on antimicrobial peptides and microbiology. We are dealing with bacterial eye infections. I'm like, kuch to mil raha hai. Because you have that entire peer pressure also, walking side by side with you in your life when you know that your friends are doing great at some places and you still have zero clue. You still at that first white blank slide. You have great knowledge, you have great recommendations, but you're not there. So I was like, okay, fine, I'll start. So I joined Dr. Sanhita Roy's lab in LVPI in 18. And then I started my work in microbiology, shifting completely, leaving stem cells and regenerative medicine behind me. There I was working on antimicrobial peptide, which is S101A12. Anyways, so I was trying to figure out how this peptide can be used as an alternative therapeutic agent against bacterial keratitis, bacterial infections. We did great work. So lucky so in the second photo in 2015 when we graduated, this was in Manipal during our convocation. Just a TMI probably over here. It was on my dad's birthday and he was not able to attend my convocation because he was not well. So they always wanted next time जब तेरा convocation होगा ना तब हम जाएंगे. And I used to make fun कि next time क्या वापस मणिपाल में admission लूँ? अभी फिर से should I go back and do a masters again over there? Life makes a complete circle. I got registered under Manipal for my PhD from LVPI. So that's the 2019 picture when I had come for my registration with my guide at the Manipal campus, main campus. I was very happy that day, by the way, that, oh, this is the place from where I did my master's, yes. So during PhD, 2020, COVID struck, by the way, guys. I was working in an eye hospital. We didn't have lockdown. We were working. I got married that year. We were working. We had a lot of troubles. Chemicals, consumable items, nothing was there. As a research department in an eye hospital and with COVID, which was such a new and a very interesting question for every researcher and scientist out there, somehow the non-essential research department became the most highlighted department of the hospital at that point. And we used to work in shifts. So March 23rd, 2020, I clearly remember we had the India lockdown and till 1st April. That was the first scheduled dates of lockdown in India, right? 1st April, we get a mail from our hospital, joining back, resuming duties from 2nd April. So when the entire nation was like, chalo, abhi ghar jana hai, shanti se rehte hain abhi, pata nahi kya hoga. We were like, okay, chalo, duty pe jana hai. And let me tell you guys, LVPI has very strict rules. Our morning classes, we have morning classes, by the way, at 7 a.m. The auditorium door gets locked at 7:00:00. 5:30 punch out time. Even if you have no work, you have to be there. You might be on your own fellowship. If you're late more than 3 days in a week, your fellowship gets cut. Because it's a hospital. That point, I had all the weird expressions that I can see among you people that, how did you manage? What? Why? But standing now, I feel that discipline is necessary in your life. That timing from 7 to 5:30, no matter what. I made sure that I did all my work during that time. I knew how to manage my time, fight with others for the instruments because we had common instruments. It's not a very lavish research institute because it's a hospital. The first priority is clinics, patients. Then comes research. Anyways, so 2020, 2021, 2022, the only thing which got me going is I need to get this degree done. I need to finish this work. It's high time. I just need to finish this. But it's not like ki chalta hua ki kuch bhi kar lo, of course, because my guide was very strict with me, specifically with me. So even when I went and showed her the data, she used to be like, are you sure? I'm like, yes, this is the data, raw data. Nahi, but paper aaya hai 2 saal pehle that it has negligible effect. I'm like, okay, I'll repeat it again. A simple experiment of checking the antibacterial effect of a peptide on the bacteria. I've done it more than 50 times. And I'm not kidding. I have the raw data of every single time I've done that experiment. More than 50 times. Just to prove her that, no, this is right. I have done this. I know this is right. I agree paper is published. Fine. But I know my data. That's right. Finally, with all those things of, when I used to go to the lab and see the data and I'm like, this is not wrong. It's literally just mocking at me saying that, try, better luck next time. वो scratch card पे आता है ना, better luck next time. Half of the time, experiments data look like that to me. That they were just like, kar lo beta, kar lo. Anyways, after all of those things, 2024, graduated again from Manipal with the doctorate degree. I was happy this time. Oh, coincidentally, my father was again sick on the very same day. But this time my mom did appoint to come and attend my convocation because I said, "There's iske baad aur koi degree nahi hai. I cannot do any more coursework. ये ही आखिरी है. Attend करना है तो करो, नहीं तो नहीं, और नहीं हो पाएगा इसके बाद." Doctorate done, right? So we had that first aim: MSc, doctorate, postdoc, faculty, postdoc. My background was stem cells, genetics and stem cells. I did my PhD in vision sciences in microbiology. Did a lot of immunological work, cell biology, molecular biology, everything. But every lab I applied to for a postdoctoral position, based on my interest, "Oh, you're from the microbiology background." Fine, agreed. When I applied to a microbiology background, they were like, "But you're from the stem cells and immunology background and cell culture background. You don't fit our lab." That was the scenario. During this time, of course, I had this thought that there's something else that I need to do, something more than this. It just cannot be, you go to lab, try out different things, check out the basic of this, keep on doing. No, there has to be some value to whatever I'm doing, right? So then space, outer space, that is something which pulled my interest. I had known about space life sciences for, I think, 2 years before that, because my husband works in the space industry. So I was introduced to the space life sciences and the amazing research that has been done in global right now with respect to space life sciences. Every research that you're doing in the labs, in the cell culture rooms, they're actually being done in space with the ISS, right? So that was something I was like, I want to try this, I want to do this. I know I have knowledge, I know I can do the work, I want to try that. So that led me to join Space Medical Accelerator, which is a company in Japan, and I'm the only space life science research advisor from India as of now. So apart from the science, the wet lab. Oh no, this is not fascinating. It just sounds fascinating. Do not clap at all. Please do not. It just sounds very fascinating. Believe me, it's not that worth. So apart from every research work that I was doing, I joined this company in Japan. So I work remotely for them from India. What we do is, there are a lot of companies which want to make devices or do experiments in space, but they are from either engineering background or pharma background or some other background, or they don't know the actual protocols or the differences or the small technicalities which would make a huge difference when they have to do a research in space. With the help that I have been getting from my husband during my PhD, because he was an engineer. And I used to ask him that, I need these micro inserts. They're very expensive. My faculty will not buy. Can you 3D print it for me? And he used to 3D print. And I used to use those inserts for my experiments during PhD, by the way. So when I had that conversation of making an engineer understand what I need, why am I telling him that, no, I need it in a cross and a circle. I don't need it in a square shape. So those conversations made me understand the difference between how a biological science or a life science person would speak with respect to an engineering scientist. I used that to leverage my way into getting into Space Medical Accelerator, because then I started contacting and talking and networking with engineer people, with the companies who wanted to do research in space, because they did not have that idea. And when they want to approach pharma companies, that there's a gap between how an engineer speaks or a business person would speak and how a pharma company would take in. So that is the role which I played, which I'm still playing. Now, SMA, I was not satisfied. I was like, okay, ठीक है, life science research advisor है, ठीक है, but Japan में है, India में कोई value नहीं है. Even if I tell someone, oh wow, oh that's nice, क्या करते हो? Oh अच्छा. The end story. No one knows about it. Then I joined United Nations for Outer Space Affairs. So I'm a member for UN currently, I am also a member for the task force of UN, a webinar coordinator for the Space Global Health Programs. So therein, India has a lack of space life science, space sciences as such, right? So we have global people connecting throughout almost once in 2 months that we have meetings, and that's the point where I realized that we do not have exposure to these things at all. We are very far behind. You know, the term that every Indian hated to hear, that India is the land of snake charmers, might not be very true, but in terms of this science age, we are way behind. Space science has researched, you know, the space science research has reached a place where it's just outside of our imagination. And that I get to know when I have, you know, bachelor's people sitting across the globe telling me that they have done their summer internship in Space Center. And if I ask any of the bachelor students in India, which I do because currently I work in Mahindra University, when I ask them, they're like, what? ऐसा कर सकते हैं? क्या करना होता है मैम उसके लिए? So that's a difference. I might be wrong, but this is kind of a general idea which is over here. So then I moved to UNUSA, United Space Nations, for United Nations Office for Outer Space Affairs. So I still work with the space sector very closely in trying to make, how would your device fit over there? If you want to do a microgravity experiment, what are the protocols you need to follow? So I have to keep myself updated, because we suddenly have clients who come up and say, oh, I want to make this device, and it should measure the body rate, oxygen, blood pressure, blah, 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 of the astronauts. And I need to make sure that the material that they want to use, or the material that I suggest to them, is actually possible for them, from where they can get, and so on. So it just sounds fascinating, okay? It's too much work. Let me remind you that. 2025, I suddenly applied for a postdoctoral position, the only postdoctoral position in India that I applied to that year. And I was very clear-cut in my head that nahi karna postdoc. I am happy with my so-called space life sciences. Nahi pata, people don't know much about it, so there's not much of question. Whatever rubbish I say, they'll agree to it. Theek hai? Fine, all set. Only postdoctoral position in India that I applied to, Mahindra University. I get a call from them, are you available for a interview tomorrow? I'm like, okay, yeah. And it was in Hyderabad. So I was like, fine. Things went on good. They work on fungal pathogenesis, so I joined their lab. It's basically, an ICMR project. That the person on, I don't know if the cursor is there. Okay. Anyways. So this is Dr. Sanjeev Kumar Chaudhary. He's the main faculty PI over there in Mahindra University. And I work under him as a project research scientist on an ICMR grant on fungal pathogenesis. So microbiology again, right? Started my PhD in bacteria, did some work on fungus, currently pursuing fungal pathogenesis, as well as space life science. And recently I also joined the All India Research Scholars Association as a postdoctoral advisory committee member. I don't know what advice am I supposed to give to them, but it just seemed right that probably, I think the overall thing is that it's not a very clear-cut road which is ahead of you. Okay, so I stand today here as not a very successful, not a success story with you guys, but a person who is very purposefully and wonderfully lost, and I found my way throughout. Somehow things clicked, and I feel that this is something that I want to do. Also, I do have days when I do not want to do research, when I do not want to go to lab, don't want to see cell culture again, don't want to handle bacteria and fungus. But then, yeah, that's life. So that's been the unscripted journey of my life. Okay, so Go ahead, do whatever you want to do, but just make sure that you love it. And it's fine. There are retries in life. There are. Okay. If anyone tells you that, oh, you should not have done this, you should have done what is the regular way, I am pretty sure I would not have been invited today here standing and giving this talk. It's probably because I've done something very out of the focus, different from all my batchmates. And yeah, so that's it. Thank you, guys.
Can you just stay up for the question?
Because a lot of them were collaborative work. So I was also working with nanoparticles during that entire thing. So I do have an experience with that also. I think it's just the experience that you collect, not the final things that you have in that.
What a wonderful perspective. Thank you so much, Dr. Priyasha. We now invite questions from the audience for our question and answer session. Please go ahead.
Yeah. In your journey, I really don't understand why you didn't like the space sector to explore, and I literally little bit disagree with you that India is much behind in space. But India showed what India is, and you know that yeah, in many technology, India is much ahead. Not only space, even in defense, if you know, the
Yes.
submarine detector India now
Yes.
has in the world is came out, and whole world nobody has. Whole Indian Ocean, no one can capture. Even China also cannot come within 20 minutes. Within 1 hour, they are detecting the submarine. Even that now recently in the war, U.S. submarine they sent, and in a few hours they detected, and that US also telling the what technology India has and how they made it. So I disagree with you. We don't have, maybe the exposure of internship culture not there where Japan you have seen, because Japan is very organized, but organized doesn't mean that they are ahead of that. I little bit disagree. Because yeah, definitely our education system bit disorganized, that I agree. But why you not pursue in the space section? That is my question, because it was very,
Ma'am, I'm
to me,
still
it's
in
very
space.
interesting.
I am still working in space.
Still you are working,
I am. I am.
but why not?
No,
Why
it's
not
not.
full time? You would give.
Uh, that's something which I've kept ahead of me. It's not that, coming back to your question, it's not that I think that India is very far behind with respect to technology. What I meant is with the educational exposure for space sciences. When I said that I do know the current status of India's priority in space sector, that is more on the defense, agriculture, and technology side. And I do know that because I am again a part of it. So I've been judges as a judge panelist for the humans in space. I do work with the ISRO for a lot of their projects. So what I mean is the space life science sector, that is neglected. So when we want to do a research, so there are a few faculties in IISc Bangalore, from Gandhinagar Institute of Technology, in BITS Pilani, IIT Hyderabad. I think there will probably be 10 faculties who are working on microgravity as of now. Yes, yes. Again, the projects, the science, the research that they're doing, they are at a they are relatively at a lower level. I mean no offense when I use the word, but yes, and I'm not talking just with respect to Japan, I'm talking in global. Because when I get into the UNOSA meetings, and when I see the research that the undergrad students are doing, undergrad, like bachelor level students, they're 17, 18 years old kids, they have more knowledge about how the bacterium or how the particular material would behave in space which I do not think our generation, our students have that right now. That is the point which I want to say. Yeah. Yes, that is probably my choice of words or framing of the sentence was wrong. I'm really sorry for it. But this was what I meant, that the exposure level is literally nothing compared to the global levels of what I'm seeing day in, day out. And I'm still in space sciences. I am still in space sciences. I still love it because according to me, I feel that yes. Yeah. So right now, when we have these, even the Indian meetings with respect to the different defense or the government, Ministry of Defense, with respect to space research, we are trying to bring up the space life science as a sector. And when I say back in 2024, I was the only life science research advisor in India, and that's true. We have this Bangalore Space Expo, which is held over here, right? I think it's biannually. Uh, yes. In India, that's what I'm saying. So, working in a close relation with a startup company, which is a part of the submarine imaging detecting modality. My husband is a part, his company is the one who's doing the imaging modalities, by the way. So, working in a very close fraternity of how the technology aspect works, I do know that when we get investors in, when we get clients in, or people funding agencies, they want the data, they want the result within a year, within 6 months. Being in science, all of us know, no life science experiment, no hypothesis can be solved within 6 months. It cannot. If you want to completely prove. Pilot readings, yes. Pilot data, yes. But when you're talking about a funding agency or any company investor who's investing 10 crores, 1 billion, they don't want pilot data. I'm sorry, they do not. That's the harsh reality of how the business world works right now with respect to the R&D. If you're involved in a startup company where you want to do research, you need funding agencies to support you. You need the angel investors, the venture capitalists supporting you, those money. They don't understand the fact that, oh, if A does not work, B will work, right? In science, what we do, even if we get a null data, negative data, at least you know that this does not work. That entire thought process is not there in the business world. It's just not there. You cannot prove your point. We want our money back. That's how cruel or, I don't know, practical the business aspect works. So that is the place where we are lacking. And this is my personal opinion, again, from whatever limited interactions I've had with the global partners that I work with, or in India, or with the company that I work with, or with the students in Mahindra University when I talk to them, the bachelor students, the master's students. This is what is my take-home message right now. Again, I might be wrong. I apologize for that if my views do not align with yours, but that's the reality. It's not something that I'm saying from only one side view. Technology, defense, yes. Technology, yes. India is spending lakhs, crores, and a lot of amount revenue with anything that you come up in the space industry which has military purpose, agriculture purpose, defense purpose. Anything else, we will see. And this we will see is not a word or a phrase that I'm using. This gets written on the proposal. So that's the reality with science. And I'm not leaving space sciences. I still work with space sciences.
Here. So I actually second with your statement when it comes to from India, we don't have a proper knowledge or background when it comes to space life sciences. And as a person who's been interested in astronomy since childhood, I just like to know what is needed for me to, pursue my career in that path right now. I hope you understand my question.
You're in your first year, second year?
First year.
First year. First, figure out what you really want to do in astronomy.
And do I need physics? I'm really bad.
Again, what you really want to do. Achut was doing biology. He ended with up physics, right? Because last night he told me that, dude, I had to cut off a lot of equations and formulas from my presentation. So yes. And secondly, if you're interested in something, you will learn it. It will not be physics. It will not be like that. Believe me when I say this. Because if I could learn engineering terms, if you're interested in astrophysics, and which part of astrophysics? Because that is a huge field. It's an uncharted water that you're talking about. Anything in space. So do what you want. Figure out what are the different things in physics, in astrophysics that you want to do. Why are you interested in astrophysics? There has to be some one small, dot that you're like, I want to do this. This comes under astrophysics, so I will do astrophysics. You need to just get that dot, clear focus on that dot.
Thank you.
Thank you all for such an engaging exchange. And once again, I request our Dean to please come up on stage to present the token of appreciation to Dr. Priyasha Mishra.
What's her name?
Thank you, ma'am, and a heartfelt thank you to Dr. Priyasha as well. And now our third and final talk of the day. I request Dr. Meenal to please introduce the speaker.
Connected to Sarvani. Are you able to hear me, Sarvani? Sarvani, are you able to hear me?
Oh, yes, ma'am.
Nice to have you over for this talk. We were looking forward to it after what to say, booming talks from other seniors. Very inspiring. And to introduce Sarvani, she's our recent graduate in 2023, and she a very determined and dedicated person in terms of her work, and the way she pursued her third semester. She did a project with me in semester 3, and like how our students were asking, can I do this subject, can I pursue physics, like that. She ended up in the project when she had to do animal experiments, and one thing she didn't, it was quite a shock to her. It wouldn't have been culture work but animal work, but she did such a wonderful job on it. At the end of it, she was a master in handling animals. So a dedicated person, very focused in trying to achieve her goal of doing a PhD. And therefore, from the time I know how she was working, she said, ma'am, I want to pursue my PhD. And she is a student engineer from SRM, and then joined for the master's program in MIRM. And she did one part of a project with me, and then moved on to Delhi to pursue her other semester 4 project. And after that, she pursued very dedicated methodology of looking for a PhD, both India and abroad. And she gave in a few exams, and then otherwise had absolute dedication and focus to reach to a point of getting her admissions. And eventually she left, I think, in 2025 January, Sarvani?
Yes.
Yeah, so she got her admissions. There were a lot of paperwork, and finally she left in Jan 2025 for her PhD. So an inspiring journey. So I hand over the stage to her to talk about how she went about getting her PhD admissions. Over to you, Sarvani.
Thank you, ma'am. Hello and welcome, everyone. Thank you for giving me this opportunity to share my journey with all of you. Just to make it easy and breathable, I would like to say that with certain discipline and certain preparation, I don't think the PhD process is hard. I just want you to all to feel that you can do it. So for me, I started it early on while I was doing my second master's project. because I think the most important thing for me was to figure out if PhD is the next step for me, and if research was something I really wanted to pursue. So I assessed myself and then thought through, because I think we need a lot of determination and perseverance to go through PhD. But once I cleared myself that this is what I wanted to do, I think it made me more focused on what to do next, because contemplating if I want to do or not wasted a lot of time. And I think you could ask people, you can always talk to people and say how your journey, how your PhD journey goes, but I think everybody's journey is unique. So one thing I realized was I didn't want to ask a lot of people how their experience was, but rather I wanted to question if research was my interest. And just to give you a heads up, I think if you start early and apart from LinkedIn, I felt Twitter was a very good search engine for me, which gave a lot of insight to how, which professors there are and which universities they belong to. And also, professors directly post on Twitter if they have any vacancies or any posts available in the lab, or if they're interested in taking volunteers or giving you any opportunities. So I think that's a good way to go, and it will help you update your data and get an idea of how many universities there are. Another thing I think we should really work on is not applying randomly, but rather being more strategic about where you're applying. Because if you randomly do it, there's a high chance you may not make it because you may not be suitable for what they're expecting. But if you be a bit strategic and you see what the professor wants or what the lab wants, and if you are having the necessary skills or the interest or the motive, I think that would be highly likely a chance for you to land on an interview. And sometimes what happens is in certain countries, in European countries especially, they have a common application program. So in that case, you may not have a chance to select the exact field that you're interested. You may have to send a common application, but you could priorly prepare yourself and select 3 to 4 areas that you're interested, and then you could give a priority that this is the subject that I'm most interested and this is the least interested. So that way what happens is you have the flexibility to choose, if not this, so that way also the opportunity of getting selected increases. um, Also, to keep in mind, some universities ask you to pay an application fee and some don't. And there are so many universities that don't ask for an application fee. So, just to give you an insight, that you can definitely choose those who don't ask for an application fee. Because for those that ask for it, I think asking for a fee waiver is not a very easy process, it's very cumbersome. And so, I think if given a chance, if you're not prepared to pay the fee, you can go with those that don't ask for it, and there are many that don't. Um, Also, if you're talking to a professor directly, not approaching through an application program, it's very important to write the email properly. Don't make it too long, and also don't be too descriptive about your interests, like I like it, or don't use a lot of adjectives. It's rather worthy if you stick to the scientific part of it, why you are interested, what you could contribute, and what techniques you already know. And also don't forget to attach your CV, because I think most of us don't, and probably we don't know that we have to. Sometimes professors do ask you back saying, could you please send your CV as well? So I think whenever you're planning to ask someone something or just express your interest, you can always share your CV. And if you have any experience letter or anything like that, even that works too. And the third thing is, once you're done with your preparation part and you know the universities and everything, the third thing comes with the recommendation letters. So I think you need close to 3 to 4 recommendation letters when you're planning to apply for PhD abroad. And I think it's a good thing if you talk to professors way ahead. At least express your interest that you are interested in applying for PhD and this is the timeline that you're looking to apply. So they get some idea and you are in a communication loop with the professors. And sending them just few days before or just before the deadline would make it harder even for them. So I think if you send it at least 2 weeks before, you know, that's a good thing. And if you decide not to apply to a university, I think it's better to inform the professor that they don't need to write and send the reco letter, because sometimes it's not good for them that they work hard and they write the letter and you decide you don't want to apply. So it's very good to make the decision early on and very responsibly apply and keep the communication intact with the professor. If this is for the application process, once you applied, when you have to prepare for the interview, I think preparation is very important, because we cannot gauge what questions will be asked after all the effort that you have put into working and making all the documents together and submitting the application. If we don't prepare the work that we have done, if we are not confident what we have done, and if we cannot confidently express or if we are afraid to answer questions, I think we will not be able to make a good impression. So I think it's a good idea to rehearse. I have rehearsed my talks before in front of the mirror, no joke at all, because that gives you the confidence, and the flow is very important. If the flow is not good, it confuses the interviewer as well. They may think that you are not so confident, and you may lose that chance. So if you practice, I think it's an added advantage, and you'll sound more confident. So that's something I had worked on, and it really paid off. And also, this has happened too with me. When I got selected in an interview, sometimes the interviews can be intense. Not that I want to scare you or make you feel afraid or anything. It's just something that we can take into consideration and prepare ourselves. Questions may not come from the project or something that you've. Sometimes it could be something generic and broad. So you should be able to smartly prepare yourself without syllabus as well, because out of the box questions are also welcome, and we don't have a control over that. Also, the thing that I want to say is, if you are preparing your work, you should also spend some time understanding the professor's work as well, because in European countries, the interviews, if they're inviting you to come to their university, you will also have a one-on-one conversation with the professor as well. So if you don't know their work, you will not be able to make an interesting conversation with them. They will not be asking you questions all the time. They would expect a mature conversation with you too. So if you are aware of their work, or if you have some interesting questions to ask them, I think that would be an interesting conversation, which would also give them an idea that you have read their work and you're aware of what's going on with them. So it looked like you're more prepared, and it will also give them a chance to explain what they're doing as well. So I would say that for every professor that you're applying, you should spend some good time reading what their work is, and probably at least know something around it. So when you have an interview, you can have a great conversation. And also another simple but a thing that I did, which most people probably don't know or some know, is that the TOEFL exam that most universities ask, I know it is expensive and you have to pay a lot of money to take the test, but another thing that you could try, I don't know if all of the universities accept, but some do, is to give an MOI, that is a medium of English certificate, medium of instruction certificate that you could get from your college. If you have done undergrad and postgrad, you will have to submit from both the universities. So if that gets accepted, maybe there's a high chance you do not have to give a TOEFL exam, which means you save money. So I think this is something I thought of sharing as well, which I did. And so they did not ask for a TOEFL exam. So I think if you keep these little things in mind and then just apply ahead. Also, for every application or for every university, the application deadline will be 2 to 3 months. It's not within a month or anything like that. You have ample amount of time to prepare for each application. So if you start early, I think it's best. Don't try to keep it till the end, because sometimes you might miss out on deadlines, and incomplete applications will not be accepted. So in my understanding, time is the key. Even though I know that there are chances that sometimes we don't get accepted, there are rejections that are inevitable, but sometimes you could ask feedback. I have asked feedback, and they have gotten back to me saying that your CV or your application score is number 8, but we have more candidates who have scored number 9. So something, if you ask for a feedback, we will get a chance to improve. And many people have this thought in mind whether having publications, does it make a big added advantage to the application or not? If you have, it's great. It is always going to add. But if you don't have, that doesn't mean you are not having a chance or anything like that. So yeah, I don't think it's a deciding factor. For me, I think how you approach your application, more importantly, how you answer those questions, is what makes the big difference. That's all. If there's any questions, I'm happy to take them.
That was truly a fitting note to close our speaker session on. Thank you so much, Miss Savani, for everything you shared with us today. On a personal note, that answered a lot of questions that I had in mind. So,
That's
so if we have one last question and answer session, students, the floor is all yours.
Sar, Sarvini, how important is it to list the techniques? Because students from master's program have, array of techniques. So, and they would do it in a practical session, yes, but may not master it. And therefore, how do we segregate that list? And is it important on a CV to list your techniques, the techniques you know?
I think people have appreciated me that it's good that you mentioned what techniques, because I think my professor found it beneficial that in my CV I listed my techniques. So it was easy for her to initially plan experiments where I was able to do with the techniques that I already knew, so that, the training time, all of that, is not a lot. So, and also it gave her an idea as to what projects she can design for me or we can do it together, which will be good for me as well. So I think it's a good thing to list your techniques, and you don't have to mention whether you're proficient or anything like that, because anyway we do basic training, but at least they get an idea that we are aware of these techniques, so it helps them design experiments or lean on to plan experiments in those areas.
And how significant it would be to reapply if there are positions in the same lab, like maybe we got rejected in the first time, and if is it advisable to apply again with an enhanced maybe training or just as it is?
Um, I think we can apply, even if we are rejected, because I think sometimes the reason may not be that you're not good or the application is not good. There could be other reasons that, some other candidate may be more suitable for the project. And the next time we apply, it may not be the same professors intaking the students. There may be different batch of students. And by then, if you have added something new to your CV and you have, done some internships, I think you will still have a good chance.
Just one more question. Is your do scholarships, like you looked into Twitter for positions, but is there any site to look for scholarships for Indian students and specifically our Indian applicants?
Actually, as far as what I know is if you're applying to a university to do a full-time PhD, you are already ensured a scholarship. I don't think you have to separately apply for any scholarship. Along with your application, you will be given scholarship. Although sometimes the scholarship will be divided in two ways: scholarship that covers your tuition fees for the PhD course, and scholarship that covers your living expenses. There is a certain possibility that you could only achieve 50% scholarship, where you're only given living expenses and maybe you're not covered with your tuition fees. But I think those issues happen mostly in the US because I think there is a structure like that. But otherwise, you are given stipend. If you're selected in the program, you will be given a full scholarship. There is no conditional thing. Conditional situation could only be there for the offer letter. If they think some of your documents are missing, is when they will give you a conditional offer letter. But that would be with request that you will have to submit an incomplete document that they require. But otherwise, scholarship I think is fully ensured. If you're going to Germany and other countries, there is a portal called DAAD. So if you go into that, I think through them you can get a scholarship. Mostly successful applicants do get scholarship through that for Indians in Germany. Yeah.
Anybody else for questions? Okay. Okay, maybe we will get back to you. If they have any doubts, they can always write to us, me, you, or yeah. Thank you so much,
Yes,
Sarvani.
ma'am. Thank you so much, ma'am. Thank you for the opportunity.
Thank you. What an enriching set of conversations this has been. Sangam would be incomplete without acknowledging each and every alumni who has taken time to be with us here today. Your presence is in itself a gift to this institution and to our students. To our alumni in the audience today, you are the living proof of what this institution stands for. You are its story, its pride, and its promise. I now request our Dean, Dr. Jyoti, to please come to the stage for felicitation of the alumni present with us today. I also request all the alumni present in the audience to come to the stage and introduce themselves. Can you please step on the stage one by one? Please come to the stage. One by one, you guys can come and we can start.
Hi, I'm Akshay from.
Okay, we can start now.
Yeah, I'm Akshay from 2022 to 2024 batch. I'm currently an intern in NCBS, Raghupadingath lab. I'm Yogeshwaran from 2022 to 2024 batch. I'm right now in InSTEM working under Dr. Bhavna Muralidharan, and I'm a project associate there. I work on understanding the molecular and cellular perspective of schizophrenia in early developmental models. So we work on organoids, creating assembloids with it to study the cellular connectivity.
Hi, I'm Sneha. I'm working in a company called iStem in CCamp. So I work with retinal epithelial pigmental cells, and we are currently in our clinical to phase 2 trials. And I am in part of their IMP preparation, like the cell preparation team, and then yeah, that's all.
Hi everyone, I'm Loshni. I'm currently working as research associate at Crayon Bio, and we work on oligonucleotide-based therapeutics for rare diseases.
Hi, I'm Vivek. I'm currently JRF in Jyoti Ma'am's lab, 23-25 batch. So I'm
Hi, I also did my master's here, like 2025 batch, and I was JRF and now a PhD. Thank you.
Hi, I'm Lauren, and I am a 2024 alumni, and I was under Dr. Jyoti ma'am. First of all, thank you so much for the talks. It was really inspiring and realistic. And so one thing I could relate was I also joined 1 month after the actual course started, and I was also anxious as to how will I cope up with the subjects, the practicals, which I lost for 1 month. But the teachers were very considerate. They asked me to stay back after classes and repeated the whole experiments again for me and few of the other students who joined late. So thank you. I'm really grateful for that. Thank you so much. And that is really helping me right now. So after masters, I have been preparing for CSIR, and I qualified for CSIR for PhD eligibility. And then I've been engaging myself in conferences, and I've also been using media for translating the concepts of stem cells to the general public. And I've also been passionate about music and calligraphy, so I've been pursuing that simultaneously. So right now I am actually looking for PhD abroad, as I recently graduated. So, and it was really inspiring to hear all of this from you. Thank you.
Lauren, and Ahana both helped us fetch cups in Rubaru. I can say that from the past 2 years. So they are the talented singers and dancers from MIRM.
Namaste everyone. I am Ahana and I'm from 2024 batch as well. And my job has nothing to do with what I did here. So I'm a full-time artist, a dancer and a choreographer right now. And I am still looking to come back, maybe eventually. But like for now, I'm gonna be a full-time artist. Thank you.
And I really thank her because for every Rubaru, without a single penny, without any support, she comes and actually trains all our juniors for participating. I really thank you for that. So, that tells the umbilical cord is never gone with our students. Yeah, and thanks a lot for it, both of you actually. Yeah.
I'm Pradeep from 2025 batch. I did my master's project under Jyoti Ma'am's lab only. And currently I am working as an intern trainee in Crayon Bio India. So I wanted to thank Minal Ma'am to initiate this great opportunity, and thanks for those talks. And it was really helpful. Thank you.
Hi, I'm Esther Rachel from Manipal Institute. I did my master's here under Shagufta Parmeen. And right now I'm working as a research trainee in Crayon Bio. And it's great to see the other side of the master. So although we had all our challenges, right now it feels okay. Okay, there are challenges in world, and it feels nice to be seeing that.
The CEO of Crayon Bio actually came and talked to me, and she was asking for people who would be interested in jobs because she's so happy with our students that they're trained well, and that's where she has asked me personally. I have actually written to Lauren, but anybody who wants jobs in Crayon Bio, they have openings. You should actually, send the CVs across. I will send to the person who is heading the scientific team there. So that tells a lot about, our students are making them proud. That's why they're coming back, and then they want more students from us, right? Yeah, the last page. Yeah. So we have, actually we are structuring a lot of things. Like now we have an alumni association, so we also have a placement, a structured placement initiative, which is being headed by Dr. Sangeeta. So we started this initiative last time, and we had some of the interviewers from IKP Knowledge Park who had come and interviewed our students. So this time again we want to have this initiative going. So Dr. Sangeeta had actually started a group, a placement group, where people there she posts new job initiatives and but I guess, how much ever you structure, like what Priyasha says, ultimately you will find your calling after all the labyrinthous routes which you follow. And most of them, they get back to research somehow. I can see that. Yeah, I think the bug never leaves you. Yeah. So this is one more thing which we want, we should make a community. If there are job posting like what Sharvani was saying, that there might be a neighboring lab which might be looking for a position, you can always put it up in the group so that the students get prepared to read the research work and then approach, right? So sometimes you don't know what opportunities exist. So if you can put a word in the group, we should structure it, Meenal, and along with Dr. Sangeeta, and we'll try how we can have the connect going. Yeah. Why I gave her 2, is it? She's not introduced. Okay, sorry. It's okay.
So, good evening. Myself Nityashri. I'm from 2025 batch. I worked in JP Ma'am's lab. And right now, I'm a trainee scientist at Cryon Bio. We work with ASOs. So, I want to thank JP Ma'am for the 1 year. All the things, whatever I learned today, wherever I'm standing now, it's because of her. She always pushes me, even if I get negative results. She always somehow pushes me, and the way she thinks and questions me, it somehow it is the reason behind it right now. And thank you so much. I want to thank Meenal ma'am as well. Thank you for organizing this, and thank you all my seniors. Thank you so much for the opportunity.
I'm the last. Shoot. Okay, come. Okay, so my name is Rishika, and I am a 2025. So they are all my batchmates. So I just wanted to say, yes, I am still currently looking into the job market. I still haven't found my placing. So currently, 2015, Priyasha ma'am is current me right now, honestly. Left graduation with a blank, and I'm still trying to figure out what is my calling. Honestly, I was paying attention to your presentation so much because it's so relatable. Yeah. Yeah, so I'm looking for current any research opportunities or for going abroad. So yeah, that's pretty much it. Also, this was an amazing ceremony. Thank you so much for having this wonderful opportunity. I never would have gotten to meet everyone like this. So yeah.
Thanks for coming.
Can we have everybody on stage for a group photo? Meenal ma'am and Sangeeta ma'am as well. No, we'll take one with the alumni first. Okay. Can we have all of you here?
It's a huge effort from Meenal and the coordinating team. It was completely her initiative. She reached out to the alums. She did everything. The only thing which I did was sign. So I think a huge round of applause to her. Yeah.
Can we also have Dr. Achyuta and Dr. Priyasha over here for the picture?
And special thanks to Harish. I think last moment, I think without him, nothing can be done. And he has done it at everything in the last moment. So I'm really, thanks Harish. Yeah. I think all of you, all the students know how important Harish is, right, for the MIRM.
We have an icebreaker session for you all, but before that, every great gathering. Oh yeah, you guys can go back to your seats. Every great gathering must come to a close, but before we do, we have one final and very important part of the program. I now invite Dr. Meenal to the stage to deliver the vote of thanks.
So thank you so much for inviting me for the vote of thanks. I want to start with how MIRM was initiated by MAHE. And the thought was like how Dr. Jyoti was telling, to bring about skilled researchers in the field of stem cell technology, and more importantly in therapeutics. So therefore the institute was established in 2007, and as a school, like, you know, Manipal School of Regenerative Medicine, and then we moved to become an institute, and then we moved here to a big university. So we've established from 2007, and we have so many students that have made us proud in almost all the technological fields, with industrial exposure and across the world. So in the United States to Europe, to many PhDs and postdoctoral researchers, and many of them spread across to the industries. So therefore we have a huge cohort of our students across the world have made us so proud. So this initiative is not mine. Getting in touch with all our students, and it was a push from MAHE. MAHE is one of the biggest interests of MAHE is to be connected with their students. So we have a committee here in Bangalore as well who drives this alumni association, and each institute we are supposed to involve the present students as a committee and the alumni together, and we are to include them in all our activities. Okay, so have an active participation, and therefore we were really inspired by their work, how MAHE in Manipal has established themselves. They have collaborations, they have interactions, they have sports, they have people having exchange programs, multiple activities. So therefore we extended same to MIRM, and therefore we started Sangam, and it's a fantastic initiative that was also guided by our Pro-VC, Professor Madhu Veeraraghavan, and our additional registrar, Dr. Raghavendra Prabhu, and Dr. Rashmi from MIT heads this consort, and therefore we are all under her managing the entire system. So we are looking forward to a lot of interactions. So the juniors as well as the alumni are, we look forward to your participation. Okay. And I like to thank Dr. Jyoti for encouraging, and a last minute transition for her. I used to just barge into her room and say, okay, I want approval for this, but this is okay and this is okay, and just would walk out. So she just said, yeah, okay. So, you know, it was like quite fast and quick, and now I think she, you know, God, next time I'm going to just organize these events. We need them, like how Achyut and Priyasha made time. We need them to be here in person, but if not, we are planning to have them online also, the interactions. So thank you so much, Dr. Jyoti. And next I want to thank our alumni, all of them who came to make an effort to, you know, Saturday, and everybody will think, oh, it's a weekend. But yet we have so many of our students that came, and especially Achyut, Priyasha and Sarvani, who took time. And, you know, it was a short notice and he was traveling, so, you know, to prepare for the presentation on Saturday, and we had so many plans, and then everybody took time. There are so many who've joined online, and they said they will, there are a couple of students who are unwell, and some just traveled and didn't know this event was going to go. I mean, they had already travel plans, so they were planning to come. So I want to acknowledge and thank all of them for accepting the invite, and those of you who came, especially the speakers, thank you very much. And I want to thank our association and the volunteers, the president, Sneha, and and Pranav, who spearheaded the entire show, and Evelyn and Purvi, who performed, and our MC, our Avantika, and Anuradha, who coordinated with Sarvani, and everybody else who really worked towards it. It was day and night actually, because they had to pull in so much in a short while. And yeah, thank you one and all. And you know, as students we do, I coordinated the phone, but the base level, if we have something really steady, is because of our admin's head, Harish, and Kumar and Marappa, of course. But then, you know, everything goes on well. I just have to coordinate, these guys will tell, and everything sets in place, and we walk into the hall, and there's juice, and then there's our snack, and you know, everything is ready. And yeah, he most of the time chooses the menu and everything is good, yeah. So thank you so much for organizing everything. Yeah. So we had Dr. Sangeeta, Dr. Vinay and others. They're joining us online, the ones who couldn't make it on Saturday. Anujit sir sent his regards, and he was traveling. He had already travel plans, so not everybody could make it. But then yes, we started. We have Sangam 2026. We want to have many more of these gatherings. So looking forward to a happy and a future prosperous Sangam. Thank you.
Okay, so I know you all are waiting for the refreshments, but we have a small icebreaker session planned. And so we all know that the best conversations happen when the walls come down, and what better way to do that than a little friendly competition. So before we head for refreshments, we have a special icebreaker session lined up. So we have a shortage of time. Yes. So we have a visual science quest over here. And whoever wants to answer can raise their hands and we'll get the mic to you. You can answer. And please